Thank you, Grace, and good morning, modelers. Good morning, everyone. I want to add my thanks to the organizers, specifically noting the leadership over the years of Chris Kelly and Grace, and your own Grace Peng, I would say. She's certainly been a champion of this activity. And to the members of the consortium who've come here, and to the members of IMAG, who actually led organizing this along with Grace and Chris. The 10th anniversary is quite a milestone. In fact, our own institute, NIBIB, just celebrated its 10th anniversary just under 5 years ago, and that was quite an event. So I congratulate you on reaching this milestone, but really want to talk to you about something that you already know, and perhaps I'm preaching to the choir, the significance of what you do and the importance that this has currently and in the future as we continue to tackle some of the greatest challenges in healthcare and the medical arena. Around the end of the scientific revolution in the 17th century, there was the creation of something that I'm trying to show you, but the slide won't come. Ah, there it is. Of something that many of you may be familiar with, some may not, this grand orrery, which actually was a model of the solar system and was used to predict the positions of the planets, the movements of the planets, and even the existence of planets before they were discovered. The planet Pluto, now referred to in a bit of a controversy as a dwarf planet, since about a decade ago, its existence was predicted by one of these grand orreries. And you can see two versions there, both of them from the 1700s, so one a bit more sophisticated than the other. So as early as the 1700s, the value and the importance of modeling, helping us to better understand our current observations, and not only help us understand our current observations, but predict answers to questions that we don't currently have. The IMAG group, Grace has already introduced, came together around 2003, these different agencies, because of the appreciation of the value and the need for modeling and how it could help all of the agencies achieve their missions. In 2003 or thereabouts, this paper penned by one of your members that you're all familiar with, Peter Hunter and his colleagues, laid out the scale of the problem and actually proposed creating a human physiome model spanning the entire scale of life from genes to the human being, the whole body. And also, not only across all of the physical scales, but also the time scales, and that's laid out in the bottom. These 15 orders of magnitude in a time scale, looking at the events that we like to observe, understand, model, predict, and use this understanding to better the human condition. Progressing all the way to population levels now with models is the goal. The timeline of key events is shown here. Grace has already talked about this. IMAG being created in 2003, issuing initiatives from multiple agencies as listed there. And then just a decade or so ago, formation of the MSM consortium, which you comprise, then continuing on with additional program announcements for support of research initiatives as listed in the subsequent time points shown on this slide, ending just a couple years ago with the one on predictive multiscale models for biomedical, biological, and behavioral and environmental and clinical systems. Truly integrative, truly understanding and supporting the need for this convergent approach to life as we know it, and similarly underscoring the importance of what you do, or the 14 grand challenges as articulated by the National Academy of Engineering, a pen about a decade or so ago, around 2007 or so. And the ones on the left point to areas that can be potentiated by models. I think these are certainly straightforward, and models will and do play a key role in addressing these grand challenges. Here is a view of Cape Canaveral, and we pull back and ascend into space from Cape Canaveral, from this vantage point of the far side of the moon, and ask a rather provocative question, noting that the far side of the moon is the furthest a human has ever traveled. And that was almost 5 decades ago. And asking, isn't it time we move forward? What stands in the way of us moving forward and achieving this goal of going into deep space and realizing deep space travel? The 100-year Starship, you see the URL shown just below the logo there, sponsored an event called the Crucible, the 100-Starship Crucible, in which a group of scientists from around the world gathered to begin discussing creating a virtual human model to sustain vitality of humans who would undertake travel into deep space, and travel that would last for years. So we know that a round trip to Mars, which is planned about 20 years from now, will take, depending on the time of the year, around 5 years or so. How do we support human life on a flight like that? How can we model the kinds of exposures that man would have in deep space travel, not only physiologically, but sociologically and emotionally, as was pointed out in the previous slide. I am particularly fond of this statement from the 100-year Starship group, led by former astronaut Mae Jemison, that pursuing an extraordinary tomorrow, which is what doing this is, will build a better world today. And indeed, that is the case. And indeed, it's clear that one of the first things to do this in a straightforward and make sense and logical way is to have a model that would allow us to tackle some of the challenges that will be faced by human travel. Here's a group of scientists sitting around in a very remote location in Austria, actually in an old castle. You can see that the view of it to the far left, but in a room here discussing the design elements that should be included in such a model and how to go about this. So I think it is clear to all of us that models are critical for accelerating progress and meeting the grand challenges that we face on the globe today as we try to improve the human condition. A charge to you as modelers: make your models useful and understandable. Quite straightforward, critical, but worth seeing. Usable by others. We are in the era of big data and big science, and one of the great hopes that we have in this era is that all of the data that so many of us in the scientific field are creating will lead to accelerated discoveries and solutions of problems by sharing the data and using the data, and we certainly need models in order to give us a logical framework for understanding what this data really tells us and for being able to go beyond what we understand now into those things that we do not understand and providing such solutions. Scalability is an issue, so we can look at questions across multiple scales, suitable for big science, clear an area, and certainly that your models should have an impact is always a goal, and a goal that we clearly discuss and is a North Star guide point for us here at the NIH. Leave you with this observation from the NAS support about mathematical sciences, you see listed here, noting that the mathematical sciences, which are critical to modeling, have an exciting opportunity to solidify their role as a linchpin of 21st century research and technology. So we look to you to provide that critical linchpin for 21st century research and technology, and to help us do what we know models do, and to put it simply, they really express the laws of nature. And that is what we're after. Thank you for your attention.
Dr. Pettigrew. Are there any comments or questions for Dr. Pettigrew? If not, I will introduce the Theme 1 moderators, Dr. Jennifer Couch from NCI, Dr. Barbara Sorkin from the Office of the Director, Office of Prevention, and Dr. Michelle Grimm from the NSF.
Good morning, everybody. Thank you for the introductions and that nice setting of the stage there. So in this first session, we're going to take a little bit of a sort of historical perspective, but also kind of a future-looking perspective. Trying to set the stage for the rest of the meeting here. And honestly, I cannot believe it's been 10 years. I still remember us all sitting in a much smaller group talking about this 10 years ago, and it seems like last year. But quite a lot has happened, and we're going to hear about that in this session and then in the others. So in this session, we're going to sort of look at the evolution of multiscale modeling over the last several years and the impact that IMAG has had on that evolution, and then look ahead to sort of think about what are the big challenges coming up that this group can help address in the next 10 years. And so we have 3 speakers in this session, and I'm going to introduce the first one, and then my co-leads are going to introduce the subsequent speakers, and then we'll have a sort of a Q&A panel here at the end. So our first speaker is Andrew McCulloch, who is both a professor of bioengineering and medicine at UCSD. And Andrew, I'm not going to go into your long and distinguished biography because I think it's going to come out as you speak, and I think this group knows you as well. But Andrew's one of those early people that had really thought about bringing together mathematics and bioengineering and biology and medicine into one package, and he's been with IMAG since the start. He focuses on multiscale modeling of cardiovascular system, and has been with us since the get-go for IMAG, so he's the perfect opening speaker.
Thank you very much, Jennifer, and being the first speaker in this 10th anniversary meeting is a big honor. So before I start, I would just like to use this opportunity to thank Jennifer and all the IMAG program officers who have supported this Multiscale Modeling Consortium, but especially to, on behalf of all of the Multiscale Modeling Consortium members, to thank Grace Peng for her enthusiastic advocacy and her very wonderful leadership of this consortium, for your gentle cajoling of the members to participate constantly throughout the year and not only at annual meetings, because without your hard work and leadership, a program like this simply wouldn't have survived 10 years, much less have grown into the impactful group that it is today. So please join me in thanking Grace. As Jennifer mentioned, I was at the first MSM Consortium meeting at the NSF in 2007, and I remember it was a cold day, Grace was pregnant, and we all fit around one conference table at that time. And I was able to find that presentation and reflected on a little bit in choosing the topics I wanted to mention today on our work on multiscale modeling of the heart. I do have to disclose that I'm a co-founder of a company that has licensed some of the software that has been developed of this research. So in looking at that presentation 10 years ago and asking where have we come and what have been the main developments and where are we going, I've identified three main trends. The first is extending models to smaller spatial scales and shorter temporal scales, particularly using Markov models as a way of bridging molecular models to cell models. But also, and this is a topic of one of the current UO1s, extending models to longer timescales, particularly the timescales of ventricular growth remodeling and disease progression. And then finally, one of the 5 program grants that I've had in the time of this program was really on extending models from the laboratory to the clinic. And so I'll talk a little bit about patient-specific models and where I think those are going. So this was a slide that I showed in that presentation in 2007, and Colleen Clancy, who's one of the new UO1 recipients, had by that time already developed Markov state models based on single-channel recordings of ion channel activity, and we were using them to integrate them into whole cell and ventricular models to understand arrhythmia mechanisms with a new level of detail. What's changed since then is that we're now making use of atomistic molecular dynamics and molecular electrostatics models, as well as rigid body Brownian dynamics models to inform these Markov state models with models instead of experimental data, so that we can identify thermodynamic and kinetic parameters of the Markov models that previously were inaccessible. And the future, really, that we have not yet achieved is to be able to make a model starting with an atomistic model of a molecular alteration or mutation and predict the whole system phenotypes using multiscale modeling strategies like Peter Hunter and others have outlined over the years. Right now, one of our interests has been in modeling the thin filament of the sarcomere, and in particular mutations in troponin C, which is the molecule that calcium binds to activate contraction. And in this particular study, we used a combination of molecular dynamics and Brownian dynamics to identify parameters of this Markov state model and the changes associated with 2 gene mutations. One was associated with a loss of function, a decrease in myofilament calcium sensitivity, and the other was associated with a gain of function. What we discovered from this is the loss of function mutation could be explained by the molecular models of the decreased binding association of calcium to the thin filament, whereas the gain-of-function mutation could only be partly explained. And that led us to explore the role of troponin C, troponin I interactions in that mutation as well. Where we're going next, with Jim Battingthwaite's UO1, working with Mike Rainier and Dan Beard and Jim, we are now turning our attention to the thick filament, because Jim's grant is about energetics, and it's the cycling of the cross bridges that's the key sort of energy-using part of the cardiac cycle. And one particular project involves looking at the effects of deoxy-ATP as possibly a therapeutic for heart failure. So Mike Rainier has worked with us on molecular models showing, for example, that the binding of deoxy-ATP to the myosin head actually exposes more hydrophobic residues, and that this may change the electrostatics. And so we're now using Brownian dynamics and electrostatic models to see if we compute how this can affect the attachment of myosin to actin and therefore alter the cross-bridge cycling. And the reason that this is a multiscale problem is because it only takes an increase of about 0.5% deoxy-ATP to ATP in the cell to about 2% to have a significant effect on contractility, and that can only be possible through the nearest neighbor interactions along the myofilament, through the cooperative interactions that involve the entire sarcomere. And then in turn, deoxy-ATP could affect the energetics of many other events in the whole cell, so we have to account for them as well. This was my title slide from that meeting in 2007. Tom Borg, who was the co-author of that review article in Nature with Peter Hunter that Dr. Pettigrew showed, was actually the co-PI of that grant. And the particular focus was on developing models that scaled from genotype to phenotype of the electromechanics of the mouse heart. But the particular result that I chose to highlight in my title slide was this paper that we published with Roger Chen, Jeff Sorserman in 2006, in which we used a model of excitation-contraction coupling in the myocyte combined with regulation, the pathways involved in beta-adrenergic regulation of contractility, and then developed a spatially distributed version of that model in which cyclic AMP could diffuse, and then using Les Low's virtual cell tool, compared our ability to model a gradient of protein kinase A activation along the cell by uncaging cyclic AMP experimentally and using a threat sensor of pKa activity. So it was nice that the model could predict this result, and it gave us insight into compartmentation mechanisms. And so what we wanted to do was take this original model that we had first published in 2003 and extend it to include compartmentation of cyclic AMP signaling. And that turned out to be much more difficult than we thought. Because the prevailing hypothesis has been that the mechanism of compartmentation of pKa signaling is mainly due to gradients of cyclic AMP in the cell. And so we made a detailed model of the subcellular space where we knew that there was a localization of cyclic AMP signaling. So this shows an electron tomogram of the small region of the cardiac myocyte, where the green is part of the T-tubule membrane, and the yellow here is the sarcoplasmic reticulum membrane. Down below is a model of a calcium spark. John Lederer sitting in the front, an expert on discoverer of this phenomenon. And then we used this three-dimensional model to see if we could also explain diffusion-mediated gradients of cyclic AMP and pKa activation in the cell. And the long story was, the short version of the long story was that we could not, unless we used unrealistically low diffusion coefficients of cyclic AMP and unrealistically high ratios of phosphodiesterases to adenyl cyclase. In other words, something else is required in order to explain the compartmentation of pKa signaling. It almost certainly has to do with the role of A kinase anchoring proteins. I mention this because it led us in a whole new direction. I had imagined we were going to make a more detailed model of the cell with more compartmentation, more compartments. Instead, we made a more detailed model of protein kinase A. And so the student that followed Jeff Sorserman, Britton Boris, made the model on the right of a Markov state model of the activation of protein kinase A. And I won't go into all the details of it, But this model turned out to reconcile some long-standing debates in the literature about the enzymatic action or mechanisms of PKA activation. Well, we then collaborated with our colleagues in the National Biomedical Computation Resource, Romeo Morrow and Andy McCammon, to use molecular dynamics and Brownian dynamics in combination using a milestoning approach. So we could take advantage of the computational speed of the rigid body approximations of Brownian dynamics, and then when the binding event of cyclic AMP happened, we could then do molecular dynamics. And so this was able then to give us new kinetic and thermodynamic parameters that enabled us to further extend this model of PKA from a steady state model to a kinetic model. So we're now doing the experiments to test some predictions of this model that may completely upend our understanding of how phosphodiesterases give rise to compartmentation of cyclic AMP signaling in cardiac myocytes and other cells. That's a little bit about extending spatial scales and temporal to smaller temporal and spatial scales. This is another slide from 2007 that was in that presentation I gave at NSF. And what it shows is something that you'd think by 2007 we already would have figured out, namely how to couple our finite element models of the mechanics of the heart to relatively simple closed loop lump parameter models of the circulation. But actually at that time nobody had done it, and I think the reason was the algorithm, which is shown on the right, that was required to make this process actually converge was a little more complicated than one might imagine. But now with this algorithm, we're able to do an important thing. You know, going from one beat to several beats isn't that interesting, but it allowed us to do something else that also hadn't been done back then and is now a big topic. So way, way before 2007, we had published a theoretical paper on the continuum mechanics of soft tissue growth and remodeling. This obscure little article, I think, became one of the starting points for the now flourishing field of the modeling of mechanics of growth and remodeling that people like Jay Humphrey have taken far, far further than this. But it allowed Roy Kerkhoffs to, in the middle of this period, around 2012, to develop models based on experiments in isolated myocytes, in which we found if we stretch the myocytes, they get longer, and in which, from indirect evidence, we deduced that if we stretch them perpendicular to their axis, they may get fatter. So using this single rule, and then coupling models using this rule to either models of aortic stenosis causing an increase in arterial blood pressure, or mitral valve regurgitation causing an increase in filling pressure, this same rule was able to explain, now this is what you're seeing here is growth and remodeling of the heart over a period of weeks, and you can see that the aortic stenosis case leads to a thickening of the wall, just as is seen in pressure overload in humans and animals, whereas the mitral valve regurgitation case, where the filling pressure on the heart is increased, causes the heart to dilate. And what we're seeing in the color coding here is the residual stresses that form in the heart as a result of that growth and remodeling. So where are we now in this? Well, those models are purely empirical. What we want to be able to do is to include more of the underlying mechanisms of mechanotransduction in cardiac myocytes. And so this is a model that we've developed with Jeff Sorselman, my former student, and his student, Philip Tan, at UVA, of mechanical signaling in cardiac myocytes in response to stretch. There are 9 mechanoreceptors in this model, 65 signaling molecules from about 5 or 6 different signaling pathways, 11 transcription factors, and what's not shown in the diagram is 645 target genes, except for these ones at the bottom that are also nodes of the model itself, and so there are translation reactions connected to those genes that feed back and modify the model. This is a model based on about 115 published papers, and in order to validate it, we've done RNA-seq experiments in myocytes subjected to stretch for different amounts of time and in different directions. And I don't have time to go through all the data, but one of the big take-homes from this particular study is the myocytes respond very differently to stretch along their axis than perpendicular, as was posited in the original growth models. And these models are able to help us identify which parts of the network are responsible for responses to longitudinal versus transverse stretch. I'm not even going to try and take you through this table comparing the model with the data. I just want to draw your attention, as is so often the case with modeling, to the failure of prediction. But the big failure of prediction that we see here, the green where the model and the measurements agree, the big failure of prediction is when the model predicts that a gene is differentially expressed, but the data don't show that. It's tempting to assume this is a shortcoming of the model, as it very well may be. However, it turns out that there is no statistical power to the conclusion from an RNA-seq experiment with 3 replicates that a gene that was not significantly altered did not in fact change. And so I think one of the challenges moving forward is to use models in conjunction with experiments like these to discover things that statistics alone can't tell us, at least not without doing much larger numbers of experimental replicates. How do we join these cell-level models of mechanotransduction mechanisms to the tissue-level models of growth and remodeling? And how do we take into account the fact that we have to do this for the fibroblast as well? Well, Jeff Holmes' U01, Jeff's former student who's now a professor at UVA, is about using agent-based models of fibroblasts and how they proliferate, migrate, and lay down collagen to predict scar remodeling in multiscale models of post-myocardial infarction growth and remodeling. So now we will, for the future, we want to be able to combine agent-based models of multicellular interactions, network models of cell signaling pathways, and continuum models of growth and remodeling to make mechanistic predictions of the long-term changes in cardiac structure and mechanics during disease. Finally, the topic of my original NSF MSM award was genotype to phenotype predictions in the mouse. And we did actually succeed in doing this. This particular study was about predicting the effect of mutations, knock-ins of serine to alanines at 2 phosphorylation sites on the myosin regulatory light chain, scaling all the way up to the mechanics of the whole ventricle. And we were able in this study to use the mechanisms of light chain phosphorylation, the gradients of the kinase, to predict the mechanics of the mouse heart and predict a specific defect in torsion that we then confirmed experimentally with MRI. This then gave us insights into the underlying mechanisms of heart failure associated with dephosphorylation of the light chain. The reason I'm showing you this slide, though, is because we did this in the mouse. And the goal now has been to extend this, and one of the UO1s was about this, to perform patient-specific models and use them in the clinic. So we've now used CT imaging to make patient-specific anatomic models, electroanatomic mapping to help make models of the electrophysiology, SPECT imaging in patients with myocardial infarcts, and then to create an electrical model of the patient's heart, in this case, patients with left bundle branch block and electrical dyssynchrony. We then do cardiac catheterization and parameterize a mechanical model of the heart and put them together to make a patient-specific electromechanical model. So I know my time is out, so I'll just take you to the bottom line with this. We were able to predict things about the outcomes of these patients that could not be predicted by the physicians. And we also found that the predicted mechanism of action of this particular therapy is actually completely wrong. That the assumption that this therapy works by increasing efficiency is true, but in fact, the more it increased efficiency, the worse the patients did. So I'll just go to the very last point in this. We've now been able to simplify these models and reduce them to models that only require an index of electrical activity. And now we're using these models to train machine learning algorithms that we can then take directly to the clinic so that we'll be able to read the electrocardiogram and predict the outcomes of cardiac resynchronization therapy. Finally, I just want to acknowledge the contribution of all of my students, and collaborators, and particularly the PIs and collaborators on the current Euro 1 awards. Thank you very much.
I think while we get our next speaker up, we have time to take maybe one clarifying question or spend about a minute here on clarifying questions while we make the swap out. Does anybody have a, oh, good heavens, okay. Well, come on up to a mic.
Thank you, Andrew. I was wondering if your full patient-based multiscale modeling would be able to model effect of external mechanical loading, like by implanted device on the appearance of arrhythmia or similar, something like hoop stress, maybe?
So Dr. Bluestein's question was whether the patient-specific models could be used to predict the effects of an external or implanted device. I think probably the type of device you were thinking of was something like an assist device, which we have not yet done. The particular device, though, that we were modeling was a pacemaker implant for electrical resynchronization. And what I didn't really have time to show was that we were able to use these models to discover specific properties of the electrical activation sequence that correlated very strongly with whether the patient responds, which happens about 60% of the time, or doesn't respond, happens about 40% of the time. And furthermore, we think we might be able to use these models to optimize the instrument settings, specifically the ventricular delay time, so that maybe many more patients will benefit from this procedure.
All right, thank you. We'll have
I get to introduce our next speaker of the morning. Ahmet Eremir earned his degrees originally in mechanical engineering from the Middle East Technical University in Ankara, Turkey, and shifted his focus to biomechanics, first in Turkey and then at Penn State in the Department of Kinesiology. Following his graduate studies, he joined Cleveland Clinic, where he still holds an appointment, and was looking at combining in vivo, in vitro, and in silico modeling to understand the mechanical function of the foot's complex architecture. In 2007, he founded and is currently directing the Computational Biomodeling Core at the Lerner Research Institute, and he's been an active member of the MSM consortium since 2007.
Thank you very much, and it's a great pleasure to be in here. I've been involved with IMAG MSM, and my research career grew actually inside IMAG and MSM, and I can feel like, thankful enough for the opportunities that I have and interactions I had in IMAG MSM. And I'll be talking about cartridge biomechanics, and how can I get it full screen? And as in any computational modeling presentation, we should have modeling problems and computational problems. Let's see if that will work. I'm clicking everything, so, well, I'm not going to bother about it that much. I will just try to, go over it and talk. It's the background presentation anyways. So we start with a clinical problem. Osteoarthritis is essentially why we look at cartilage biomechanics, because it influences like 26.5 million only in the United States. It dramatically increases with age, and you can see in here how a healthy cartilage, with a grade 0 can degrade and essentially become catastrophic, failure, and which is laying over the bone that is like loaded, and that actually enables mobility and, as a load-bearing
tissue, and it doesn't necessarily work as well anymore. So again, it is like 33.6% of adults over the age of 65 have some form of arthritis, and mostly in the knee, and it results in a lot of knee and hip replacements. And 80% of the patients have movement limitations. And this is not the only issue that we see in cartilage. There are a lot of other cartilage disorders that is related to mechanically induced loss like tears, trauma, injuries, and defects. So how does mechanics fit into the game? And yeah, cartilage is a prominent load-bearing tissue, and its load-bearing capacity depends on the maintenance of the extracellular matrix, which is modulated by the mechanical signals going through the cells. And when the cartilage damages, it is hard to recover actually, because the cartilage doesn't have any blood supply, and if the cells are damaged, they cannot maintain the matrix, and that's an ultimate loss in the end. And aging, joint disorders, interventions can alter mechanics at many different spatial scales. And as you see, this is cartilage and osteoarthritis is an interesting problem, now reaching about 5,000 publications a year, and the mechanics is essentially constitutes about 6 to 7% of those publications, which is at a rate of 300 publications a year. So there is a load-sharing pathway. As we walk, our body is loaded, and those loads are distributed to joints, and within joints to different tissues in the knee, for example. And within the cartilage, it will be distributed to different zones of the cartilage and the cells in the different zones and the fiber structures of the zones. So this is a load-sharing pathway, the mechanical environment that goes across scales. And from the other end, from the bottom up, there is the role of structure and function. If you have a fiber-based structure at the bottom, that can dictate the macroscopic tissue response. Macroscopic tissue response can affect the joint level response, and at the end, the body level response. And that bottom-up process essentially dictates mechanical capacity at all those spatial scales. So before I get into multiscale modeling of cartilage biomechanics, I just want to remind you about the charge that we were given in this presentation. And we want to talk about a historical perspective and impact of IMAG and MSM. And I will discuss some of our current work and how IMAG and MSM facilitated some work, and maybe point out some future directions. And from a structure-function perspective, this was a traditional, view of doing multiscale modeling in cartilage biomechanics. And it has a few decades of work behind it too, because cartilage has different zones. Fibers are aligned differently on the superficial zone down to the deep zone. And of course, this collagen fiber alignment can dictate depth-dependent properties, inhomogeneity, and isotropy. And this architecture is different where you look at the cartilage. Even within the tibia cartilage, medial side, lateral side may exhibit different, fiber arrangements. We have proteoglycan, we have interstitial fluids and charged particles. That gives the cartilage a really nonlinear mechanical properties at the macroscopic level. And therefore, there has been significant motivations to go from micro level to macro level modeling and simulation, because constituents and their organization indicates the macroscopic mechanical properties, and the influence of microstructure is essentially on the material properties a blueprint for tissue engineering. And tissue engineering is a very active area for the cartilage to replace essentially broken cartilage. And again, the development, growth, and maintenance of the matrix is partly driven by mechanics. And one interest, for multiscale modeling in this era, was to essentially use imaging to understand the constituents and the content of the cartilage, and maybe from that content, build some image-based models to understand the mechanics in an individualized manner too, and essentially do the mechanical assessment in an individualized manner, which may have a risk assessment value or a diagnostics value. And from this modeling perspective, the biomechanical properties at different scales are the markers for various clinical and scientific reasons. And I'm not going to go into the details of multiscale modeling on structure and function. There is a plethora of literature about that and the reviews there, and you can check in the links that I provide in the presentations to reach a few. but I'm going to be more talking about the load sharing aspect. And somehow this happens to be a void, like when I started looking at the cartilage biomechanics, how we can actually understand the influence of body level loads down at the tissue level and down at the cell level. And it has been known that joint stability and congruency of articulating surfaces are indicators of healthy cartilage mechanics and risk of cartilage degeneration. But the details of how is not necessarily known as much. So we know that there is at the joint level parameters, and joint level stability and congruency will influence the cartilage health. And of course, the activities of daily living dictates the active stabilization, like muscular loading. The other structures in the joints, ligaments and meniscus for the knee, for example, will dictate the passive stabilizers and essentially affect the loads acting on the cartilage. And at the cellular level scales, the cartilage matrix properties and the cellular organization may dictate the extracellular matrix loading, fibril loading, and as well as cellular loading, and things that are really important for the damage of to prevent the damage of cartilage and to essentially prevent the cellular damage. And of course, the motivations to go from macro level to micro level modeling are that joint loading and properties dictate cartilage and chondrocyte mechanics through this multi-scale load sharing workflow, and the mechanics plays a role in chondrocyte activity, differentiation, vitality, and matrix damage. And pathologies, injuries, and interventions may have different impacts at joint, tissue, and cell scale. So what may seem as safe at the joint level, an activity or an intervention, may not necessarily be safe at the tissue level or at the cell level. And this was more or less the motivation of how we were started to assemble musculoskeletal modeling to understand body level and joint level loading and how to feed that information through some finite element analysis studies at the joint and tissue scale and at the bottom level tissue and cell scale. And in a sense, what we are looking at now is the biomechanical response at different scales as markers for risk assessment, diagnostics, and intervention performance. And experimentation to acquire this biomechanical response is rather limited, actually, and that's why we need to rely on a multi-scale modeling strategy. So how did IMAG and MSM come to play for cartilage biomechanics? And I got involved with the multi-scale modeling consortium around 2007, and at the time, Trent Guest actually got me involved with this, and he had one of the first grants related to the cartilage biomechanics and knee biomechanics in dynamic simulations of joints. And I was fortunate to expand these explorations. At the time, we were looking at how to couple musculoskeletal movement simulations and musculoskeletal loading with finite element analysis of tissues to correlate muscular forces, joint level forces that is happening in daily activities to tissue level stresses and strains. And we wanted to take it further to go beyond the tissue down to the cell level, because that's essentially where the mechanical signal becomes really important. And we started the project on predicting cell deformation from body level mechanical loads. And in subsequent years, Kevin Shelburne from University of Denver, has started working on coupling FEA again with musculoskeletal modeling as it relates to joint replacement, which is a consequence of osteoarthritis if it cannot be prevented or taken care of by any other means. And also through Yasin Dyer's and Daryl Thielen's work on using multiscale modeling, following ACL reconstruction. And this relates to cartilage biomechanics as well, because ACL reconstruction or the lack of ACL may change the joint mechanics, and it is known that when the joint mechanics is altered, cartilage may start degenerating. So here is an example from Trent's work, and actually that was one of the initial studies that was more looking at the role of meniscus on how the meniscus may change contact load description. But one thing interesting about this study was also kind of looking at both musculoskeletal models and the finite element models and trying to calibrate the multi-body models with finite element models. And this wasn't, important because finite element analysis type is not necessarily that convenient to use looking at the activities of daily living with muscular loading and etc. at the time. And that's why simplifications need to be made, and then some of the structures need to be, put into the musculoskeletal models, albeit with lower fidelity. But this study actually showed that it was possible to incorporate meniscus into multi-body models. And in following, we started looking at the feasibility of simulations from joint scale to cell scale, and we were able to actually run a joint tissue scale model, get the tissue level stresses and strains, and figured out some multiscale data passing problems, and tested some of the assumptions, and run about thousands of cell scale models to essentially obtain figures, as you see in here, about how the effective strain at the tissue level may be related to the individual strains in the chondrocytes. And what we also realized that there was a significant lack of data, because for modeling we need anatomy, but just having anatomy doesn't necessarily mean that you can get a specimen-specific model. So we actually did a very comprehensive test on one knee, one cadaveric knee, but that test actually takes weeks with testing multiple specimens. But we did joint testing, we did tissue testing, we did tissue histology, and essentially acquired a multi-scale data set that provides joint response, individual tissue responses, and also the tissue, histological examinations to understand cellular distribution. That can actually wrap up, to build a whole multi-scale model set that are purely specimen-specific. And we also realized that we had to build models in a high throughput and run simulations in a high throughput manner, because we want to do it clinically. So if someone comes in and if we have an understanding of what the contact mechanics may be, we want to build a regional joint tissue scale model, and we want to do it fast, so parameterization helps in here. And once we run it, we can actually get the multiphasic, mechanical response from those models and feed it to cell scale models that can be built very quickly based on potentially individualized cellular anatomy, cellular organization, and the material properties, and run simulations such that we can relate the contact mechanics or joint mechanics down to the level of cells in an expedited manner. And on the our colleagues in IMAG MSM has been working on more on the body and musculoskeletal level. And Kevin Shelburne, for example, and his team provided a way to individualize the knee joint models. And one problem is always getting the ligament properties right, because if your ligament properties, which are passive stabilizers, are not that right, your knee joint mechanical responses will be erroneous, or maybe it may not be that erroneous, but maybe may not be at an accuracy that you may like. And if the knee joint kinematics is not that right, then that will also reflect to the contact. And he essentially devised the methodology to use specimen-specific joint mechanics data to fine-tune those ligament properties and to calibrate those. And that may not be possible, of course, because sometimes we may not have that level of data, particularly for human subjects. But a work by Daryl Thielen and Yasin Daur's project essentially did high-throughput modeling and simulation and Monte Carlo analysis to investigate the importance of ligament properties, not only on the joint kinetics, but also on the contact. And here is a good example too. And of course, one way you can look at it as an uncertainty problem, how we should model the ligaments to get the appropriate contact descriptors in here. This is a contact parameters influenced by the anterior cruciate ligament stiffness and its reference strain. But you can also look at it from a clinical perspective too, because if your ACL is ruptured, then your ACL stiffness is essentially 0. Or if you put a reconstruct an ACL, the stiffness may be different, and that might affect the contact in the cartilage. And so in summary, all those MSM projects delivered new knowledge on cartilage biomechanics as a function of musculoskeletal activity, and we got high throughput so that we can couple joint loads to contact mechanics, to zonal mechanics, to control site mechanics. We documented some of the uncertainties. We illustrated the individual possibility to individualize models. And most importantly, we made data and models available. And now, someone who wants to get into knee joint modeling and cartilage modeling can actually exploit these data and the models and doesn't necessarily reinvent the wheel. And of course, in the world, there were intensive research activities. Again, remember, there were about 5,000 papers in cartilage coming out, 300 papers in cartilage mechanics coming out. And there are some of the projects that are illustrated in here. I'm not going to go in details, but all of those projects provide significant amounts of data from clinical to scientific levels, and that can be utilized to look at the cartilage biomechanics, knee biomechanics, joint mechanics in its entirety. I also wanted to mention a few other activities that I was involved with the working groups, and this was really important because these activities really changed my way of thinking, and I was involved with different working groups as you see in here, but we were able to, a cartilage biomechanics and the lung biomechanics can, start writing, and discussing ideas and challenges about multiscale modeling and computational biomechanics and write a white paper about that. Or, one thing that we did as part of the, biomechanics working group was to identify reporting considerations and reporting guidance, like for finite element analysis. We ended up finding about 80 parameters that are important, reporting parameters that are important. And the Committee of Credible Practice of Modeling and Simulation in healthcare really opened my eyes to the different worlds, different domains in modeling and simulation, and how we can come up with a unified approach for credibility. And you can all see some examples of these in the posters. And most recently, we also tried to see how model sharing and reproducibility can be integrated to scholarly publishing. And again, the modeling and simulation world is going on, and while we are also working, and we were exposed to those activities in IMAG and MSM too. And you have the presentation, you can follow all those links there. So I just wanted to wrap up. So where are we now? Now the good thing is we are not blind anymore. We can look at cartilage biomechanics in its entirety, and that is really important. And we are hoping to have a handshake between mechanics and biology, biochemical markers, imaging markers, biomechanical markers, and essentially clinical outcomes. And we will do that with multiscale modeling, because that will essentially give us the opportunity to understand why some associations that we find in big data are there. But we also need to, reach out and use some of the big data approaches to understand voluminous literature, to understand data from activity, and maybe to consolidate our data. Because we want to get individualized and actionable knowledge to guide scientific discovery, innovation, and clinical care in an accessible and credible manner. So I just put it in here, the mission of IMAG MSM and the opportunities, and I'm not going to go in detail. It's there. Let's sustain the mission. Let's establish a synergy nationwide and internationally, diversify our funding opportunities, expand our community, and us in here, let's exploit all those opportunities that is given us by all the IMAG members that I'm very thankful. And thank you very much.
All right, so there we go. It actually does work. Grace has asked me to request that everybody start putting their questions on the wiki so that they can be gathered for the panel discussion, and then we will bring up our next speaker.
I'm not the next speaker. I am happy to say that we are now going to, in keeping with multiscale modeling, Dr. Pettigrew, what Ahmet just talked about, we are going to zoom out and take the scale from the molecular to the population level. It's a pleasure for me to introduce Cecile Viboud. Cecile received her degree in biomathematics from the Université Pierre-Marie Curie, and she has been, since she received her degree, at the Fogarty International Center here at the NIH, and I will let her tell you about her work.
Good morning, everyone. And thank you to Barbara and great for inviting me. I'm a bit of an outsider to this group, I guess. But in my world, which is infectious disease modeling, we also think about multiscale modeling, and in particular, spatial transmission models, which is what I want to talk about today. So the history of spatial diffusion models for infectious diseases started in the 1960s in Russia, and a couple of Russian scientists thought about building metapopulation models where you think of different cities that might be connected by population movements, such as trains, for instance, in the 1960s, and how diseases might diffuse through this network of cities. And then an American scientist, Ira Loncini, started collaborating with them, and is now still working in this, but brought by the technology to the US, and it has flourished since then. In the 1990s and early 2000s, there was a shift and an effort to think about agent-based models, a very detailed model of the entire population of a country or even the world, and to use those models to model the invasion wave of new pathogens. The issue, however, with such great detail in the modeling was that it was very difficult to calibrate against empirical disease data and make sure that the models were true. And so our approach has been to start with the disease data that's available and build models from those data, which is what I want to tell you about today. So where were we 10 years ago? So we only had access to very limited disease data, and here you see a typical time series of influenza incidences on a weekly basis for the US as a whole. Flu is a very good model system because there's a lot of data available for flu. It's an interesting system for its evolutionary dynamics as well as epidemiological impact. So it's usually the models get developed for flu and then translated to other pathogens. So from those very coarse, nationally available time series, we could break them down by state, but we could not get down to a further level of detail. But in those very coarse data, there was a signal that that the diffusion of the disease across U.S. states was driven by workflow or the movements of people moving from home to work on a daily basis. And the traditional model to model population workflows is this gravity model, which comes from physics, and which, as for ingredients, population size of the donor location, population size of the recipient location, divided by distance. And there are exponents to those factors that you can estimate from this data. And we could estimate those from the US flu data and then come up with scenarios and prediction models for how a new wave of influenza would spread across the US. But again, very coarse data to play with. More recently, we've been able to work with a private big data warehouse system, IMS Health, which records visits for flu-like illness across the U.S. It covers about 50% of U.S. physicians, 70% of the U.S. population, so a very powerful system. And through this, we can monitor how the disease activity in the different cities and correlate that with census information as well as weather factors. So the first thing to do is to make sure that the data, the disease data that we have at hand makes sense. And so we validated this data against a number of traditional disease surveillance systems in the US and at a different spatial scale. And here we're looking at a local scale, so New York City, which is a very good test system because they have lots of health data there. So in the middle here, you see their gold standard data based on their emergency department visits. So we have flu-like illness for a couple of years, and here in 2009 you see a large spike associated with the emergence of the 2009 influenza pandemic, which was a large invasion wave in the US and globally. And here at the top we have this private big data warehouse system, and you can see that the two time series are very similar. When we look at correlation in weekly incidence, they're very good. When we look at correlation in timing or peak week, they're very good, or the epidemic size are also very good. So those medical claims big data warehouse system are very good to track influenza locally. In contrast, there's also been in this world a lot of interest in looking at digital data as a way to monitor flu and other disease, in particular Google Flu Trends, which is a system that tracks when and where people search for flu and other illnesses. And we've shown that actually Google flu trends was very poor, especially locally at the scale of New York City, for instance, and completely missed the first wave of the 2009 pandemic, which is a major thing to miss, a major event to miss. So in this big data medical claims system, we can also see very interesting heterogeneity in disease patterns across the U.S. Here we have, for instance, the patterns for the 2009 pandemic in California, which was marked by a very small spring wave, and then the major activity was in the fall when schools reopened. In contrast, if you look at New York State, there's about a very balanced impact of the two waves in 2009. But even if you break down the data further at the scale of cities within New York State, you see even more heterogeneity. For instance, if you look at cities, and at the big city, New York City, for instance, Bronx and Brooklyn, you see that the major impact of this pandemic was in the spring, and then there was very little going on in the fall. But in contrast, if you look at upstate New York, such as Elmira or Glen Falls, the major impact of the pandemic is in the fall, right? So lots of heterogeneity at different spatial scales here, either the state of the city. So we can look at those time series data and assign a timing of epidemic onset for each of the 300 or so cities that we have data for, and look at the timing of the pandemic across the U.S. And this is here focusing on the fall wave, which as a whole in the U.S. was the major impact. And the colors represent different timings. Green and yellow is early, and then blue is very late. And you can see there's a lot of spatial structure in this map, right? I mean, the colors are not completely mismatched. I mean, there's a lot of yellow and green here, and so showing that there's a radial diffusion of the 2009 pandemic wave out of the southeast U.S., in particular Alabama and Georgia. So there's a strong spatial structure in this epidemic, and then it spreads now throughout the north and the west of the U.S. over a 3-month period. So we can model this spatial structure and the timing of epidemic as a function of different factors. So here, if we have an infectious city that might transmit infection to a susceptible city here in green, we can think that perhaps the population size of the infectious city might matter. The population size of the susceptible recipient city might matter as well. Whether schools are open might also matter, because we know that pandemic flu is a disease that's very strong in kids, so schools might also affect the dynamics of this disease. And perhaps weather factors such as humidity, which in animal experiments has been shown to be important for influenza transmission, might also affect those spatial patterns. So if we can write the risk of influenza transmission from this infectious city into the susceptible city as a function of the sum of all those different factors and estimate them from the data. And we can see here we have a graph of the model fit for 2 different models. One model here on the left that's purely driven by schools. So you do get transmission if schools are open, but no transmission if they're not. So the red dots here are the data, the flu, the empirical disease data, and then the gray lines are the confidence intervals of the model, and the orange are the mean estimates of the model. So you can see that this model here on the left that is just school is doing very poorly, right? It's not reproducing the red dots very well. But on the right side here, if you have a model that's purely driven by distance, if 2 cities are very close together, their timing of epidemic is very similar, but if they're further apart, the timing is more dissimilar, then this distance-based model does very well. And even better, if you have distance time school, you get a better explanatory power for the diffusion of this pandemic wave in the U.S. So we've now looked back at other outbreaks of flu, not just the pandemic, which here appears on the right-hand side of this graph, but also the seasons before the pandemic. So this is what we call seasonal influenza. It's the reinvasion wave of a pathogen that we've already seen. And in the same way, we can look at the spatial structure of these seasonal epidemics. One key ingredient of the spatial models is to assign a timing of epidemic to each location, which we've designed a new method to do here with the seasonal data. So we have an example here for a small city in our data set, Aberdeen in South Dakota. So this is the first of the 300 or so cities that we have data for, and this is for one of the flu seasons. And you can see here that before the epidemic, there's a noisy pattern of very low incidence, and then at some point, when flu reaches Aberdeen, South Dakota, there's a sharp rise in the number of cases over time, right? And we want to really try to measure the breakpoint between those two regimes, the pre-epidemic period and the start of the growth rate. So we use what we call a broken stick method, where we feed two piecewise linear models to those data. And you can see here that we have a very good profile likelihood for the estimates of the timing of onset of the epidemic around week 26 or 27 here in those data. So we can do this for all cities in our network and for all seasons that we have data for, and then produce maps of epidemic timing of influenza across the U.S., which you see here for this 2003-2004 winter season. And you can see that here we have a lot of cities in Texas and Louisiana that tend to be early, and then the northern part of the U.S. tends to be late in this particular epidemic. So we can do this for all seasons that we have data for and produce those maps of timing. And what's quite intriguing is that across all those seasons, we have 7 in which the flu epidemic starts in the south of the U.S. Not always in the same location, sometime in the southeast, sometime in the southwest, but it is always in the south of the U.S., which runs counter to some of the hypotheses about how low absolute humidity should drive the transmission of influenza. I'm just going to skip this slide in the interest of time. And so we can also run models, fit models to those seasonal timing data, slightly adapted from the earlier models we saw earlier, but you still have the same concept of an infectious city and a susceptible city, and there's transmission of an infection between the two. And then the risk of transmission depends on the baseline factor, as well as some measure of population size, as well as distance, plus a normalization factor that we can estimate from the data. So we can estimate those model parameters for all of the different seasons that we have data for. And without going into the details of this table, we can see that the distance exponent around 2 to 2.6 is quite constant across season, and that indicates a very sharp decay of transmission with distance. And then population size is important as well, which means that larger cities have a higher risk of infection as compared to smaller cities. And then the normalization factor is always different from 0, which tells us that it's not necessarily the number of neighboring cities that matters, but the proportion of the cities that are infected for your risk of infection into susceptible cities. We can also think of variations of this model to take into account not distance, but other mobility factors that might be important for the transmission of the disease, in particular air traffic or walk commute. And so instead of the pure geometric distance that we have between cities here, we just replace with the amount of fluxes between any two cities that depends on air traffic and then walk commute. And then we can compare how the different models fit the data. And we can see that the best model, which here on this table is listed by the smaller AIK criterion, is most always for the model that's driven by distance. That the model driven by work commutes does relatively well, and particularly in one season, is good. And the model driven by air traffic is particularly poor to explain these data. So distance is really the key factor at this scale. So we've since been able to look at the data at an even more refined scale, and instead of looking at cities, now we're looking at zip-free level data. And fitting similar models, we're interested in finding regional hubs for the transmission of the disease. And those regional hubs here are color-coded. So this is for the 2009 pandemic again, where again we have a lot of power because it's a very clear signal in the data. And you see there's a hub here in California, and in particular out of the Central Valley, Stockton here. There's another hub here in Arizona, and then there's a hub on the East Coast, and then a couple of others. But so those hub cities, which are identified by the model as being really important for the transmission in the region of flu, tend to be relatively small cities and sometimes isolated cities, which runs counter again to some of the earlier argument that flu should be transmitted across the very large cities that get a lot of air travel, for instance. But when you look at the very detailed data, that doesn't seem to pan out. So the last point that I want to make here in this presentation is that in parallel to this epidemiological data set, there's a huge amount of information also that comes from looking at genetic sequence, so the sequences of the flu virus and how that varies spatially. And this is here a tree based on a large sampling of flu sequencing that was done during the 2006-07 season. And here on this tree, the sequences are color-coded by region. And you can see here that there's a mismatch of color on this tree. And so this suggests that there's actually no spatial structure in this genetic data, which is very different from what we see in the epidemiological data, right? If you remember those maps that I showed, there was quite a bit of spatial structure there. So the spatial structure that we see in EPI, so far, we haven't been able to see it in the genetic data at the scale of cities. So this is my last slide. I'm actually a bit ahead of time. But so those spatial transmission models have been very good for us to better understand the diffusion of flu and potentially other pathogens. We have ideas for extending those to other rapidly transmitted diseases. But we've seen a lot of heterogeneity of season. Surprisingly, most of the epidemic tend to start in the south, which is very intriguing, and we want to look at longer time series to see if this is confirmed in more data. We've also seen that distance is a very strong factor, especially when you look at very detailed spatial data, the level of CT or ZIPs. And population size is important, as well as the proportion of neighbors infected. We tend to see that there's a more diffusive mode of spread in seasons that are dominated by the emergence of a new virus, such as the 2009 pandemic, or seasons that, for instance, are linked with escape from the vaccine strain. And we think that this is perhaps because those seasons have a stronger impact in children who are less mobile than adults. Overall, thinking about this model, the granularity of the disease data that you try to calibrate your model on really matters. And we really need to have more resolved disease data there, and we are really miles ahead from the weather, world where we have really great observational data. But for epidemiological and infectious diseases data, it's really an issue. And then finally, the frontier, I think, for this field will be to reconcile the spatial patterns that we see in epidemiological data with those that we see in phylogenetics or in pathogen sequence data, because right now, at the scale of a country, they just seem to be very different. And with this, I'm going to end. Thank you.
So while the other two speakers from the session, Ahmet and Andrew, are coming up to the table, are there any clarification questions for Dr. Viboud? Come to the microphone.
A quick question. So where does the flu hide for the rest of the year?
Can you hear me? Yes. So it's been a long-standing question in this field, and the answer seems to come from phylogenetic data in this case. And the idea is that it just disappears from the US and other temperate countries in the summer and hides in tropical regions where there is flu year-round as a whole, and then comes back in the next winter. So there is no sustained chance of transmission in temperate regions throughout the summer. I guess you missed all of this. No, was it good? Yeah.
And let me just remind people, if you have questions or comments, please post them on the wiki, or come up to the microphones with them if you're in the room.
I'd like to ask a question of all three of the speakers. And the backdrop for this is in the last half of the 20th century, probably one of the biggest transitions in solid mechanics was the recognition of hidden length scales being important. So this is the world of fatigue and fracture. And so when we look at strength of a structure, there's a decision. Do you need to make a multiscale model, or do you just do some sort of continuum analysis? And in solid mechanics, traditional solid mechanics, we have the framework of linear elastic fracture mechanics to determine if you need to go down and give Marcus Buehler a call for an atomistic simulation. So a question broadly for you guys is that in the bio world, how close are we to coming up with an analog to the linear theory of fraction mechanics? Andrew pointed out that it's cardiac resynchronization therapy. Turns out you can get rid of the mechanical side. And Ahmet doesn't have doesn't need careful cellular level models in his work. And I didn't catch your name, but the distinguished speaker at the end is still looking at population level data instead of individual events. How do you know if you need more?
So I think this is a wonderful question and a really hard one, because the only thing I've really been able to conclude in biology is it depends very much on the question on the specific system, and we often don't know until we try. I think in general, the strategy of building representative volume elements and putting as much detail as you can in, which can be very computationally expensive, can be illuminating in different situations. Where possible, we try and sort of jump over the need for a lot of microscale structural heterogeneity in the mechanics. But I think we can get away with that in the heart, because the mechanics of the heart at the microscale is being dominated by the contractile filaments. So all the other structures which are affecting the three-dimensional mechanics are important, but their molecular interactions are sort of secondary to the strong molecular interactions between actin and myosin. But then if I was to look at a different system, or look at a part of the heart that wasn't contractile, such as an infarct where there's collagen fibers and proteoglycans and fibroblasts, then I think the answer to the same question is different. So other than sort of using the techniques in solid mechanics has given us to try and attempt to model representative volume elements in as much detail as we can, and then homogenize and see if it works. I don't know if I can give any strong rules.
I think that's also like very, analogous to a problem that we see in specimen specificity too. How specific we should get, right? Or like how many length scales we should go. And really, there is not a solid answer to that, which relates to like what Andrew is like. If we don't do it, we don't know where our base point is. And then we can do a sensitivity analysis, and we can see how things may be affected. But in the end, what we can live with depends on the question we ask. And some questions are very suitable to simplified models, and the sensitivities of the models doesn't necessarily affect how we answer that question or the conclusions we reach. But it's more rather than looking at the absolute reality, how we can assess the useful reality. And and that depends on the question.
Yeah, very much agree. That depends on the question. There's a very strong worry in the infectious disease modeling world that we have to use parsimonious models. So, a lot of the time, to respond to your question, you don't need, the increased details, but with the advance of a very detailed agent-based model, now we can test whether some levels of details matter or not when you're thinking about large populations.
So we have 3 questions on the wiki, and we are prioritizing the wiki questions now. So Simon Khan, if you want to go to the mic, I'll ask your question, and if you have a clarifying question. Simon Khan asks, how do limits on computational capabilities of available tools most impact progress in multiscale modeling? Where would computational acceleration most benefit progress?
Well, certainly computational power is limiting us, and I think 10 years ago I would have imagined that it was in developing the multiscale models, and it's certainly true that of all the scales of modeling I showed, that it's the molecular dynamics that is the most computationally intensive. But I think today, where I'm seeking collaborations with the national labs to get as much computational power as possible, actually isn't in those very large multiscale problems. Rather, it's in the patient-specific modeling. And the reason is because the patient-specific models, which themselves are somewhat expensive, are optimizations that have to be then computed many times. But then on top of that, we want to generate hundreds of thousands of virtual patients that we can then use to train machine learning algorithms. So you put all that together, and it turns out, unexpectedly for us, that as much as the large multi-scale problems are computationally intensive, given access to the biggest computing power available, I'm choosing to use it right now for developing large libraries of patient-specific models for training machine learning algorithms.
Okay, we have two questions from Jimmy Moore. Jimmy, if you want to stand up. Both questions are for Dr. Viboud. Could the small city hubs be associated with the presence of nearby military bases? And the second question is, as in the military bases are likely hubs of transport?
Yeah, I mean that's an interesting point. We haven't thought about it, and we could look at mapping, military bases. I think the idea of incoming fluxes from outside. We would also see that in the big cities, right? There's lots of tourists getting into New York City and some of the other big airports, and that doesn't seem to drive the epidemic in the U.S. that much. We could look at military bases, looked at demographics of some of the cities, and we didn't see anything special, but we certainly haven't thought of everything. Thanks.
Can I ask a follow-up? Sorry. What about rates of vaccination? Do they affect the appearance of hubs?
Yeah, so most of the data that we've looked at was before the large-scale pediatric vaccination programs in the U.S., which started around 2006, really after the pandemic, 2010. So there is a constant, throughout the U.S., we're vaccinating our senior population quite a bit. Coverage is about 60%, but that's quite homogeneous across the U.S. There's more differences in pediatric coverage, but again, that's post what we're looking at.
Please use the mic.
Thank you. I have a question from Cecile. As I noticed, as you mentioned, hot cities are not large cities, but at least for one of them, Boulder, Colorado, I see it's a college city. Do you think that could affect being at a hub because the students distribute the disease basically by traveling to their back home or hometown or something?
Demographic is an important factor, especially for the 2009 pandemic, which was strongly focused on young segment of the population. However, this particular city is not the only college city in the U.S., and we've also looked at demographics across the U.S., whether that would predict those hubs, and it didn't. We looked at different age brackets, and so there's just no consistent pattern. What I should say is that we've also now taken this model to look at seasonal flu, and while we always see that small cities seem to serve as hubs, it's not always the same, from one season to the next. So there's also lots of variability between seasons, which is very characteristic of flu, and sometimes an annoyance, but we have to deal with it.
So there's one more posting on the wiki, but do you want to go with the audience questions?
I posted my question on the wiki, but apparently in the wrong place, so Grace didn't see it. So my question on the wiki was, I really enjoyed Ahmet's presentation, but when I think about osteoarthritis, I do not worry about chondrocytes. I worry about pain because my knees hurt. So the question is, can you link the complex multiscale mechanical problem to things that are causing pain? And as long as I've got the mic, I also want to ask the general question for all of the panelists. If you ask me what's the most exciting thing that's happened in biomedical engineering in the last decade, I would say it's the improvement in imaging technology. And I'd just be interested in your comments on how you think imaging technology over the next decade will influence the development and implementation of multiscale models of any type.
I'll start with the pain problem. And pain is actually one of the most important issues, like in osteoarthritis, and it's a holy grail of osteoarthritis too. And Now, the interesting thing is you just need to look at where the pain receptors are, and which are not necessarily in the cartilage itself. And you still need to, and I think there are like theories about if the pain comes from the bone, from the capsule, if the fat fossa has an effect on, essentially, fat fossa transmits the loads like within the tissue and then stretches the synovium, the capsule. So, and we can actually look at those kind of hypotheses and theories in a very similar modeling approaches that goes from joint level, but this time looking at what are the mechanical signals going into chondrocytes, look at the mechanical signals going into tactile sensors, pain receptors. And that can actually, we can test some of those hypotheses through the framework of multiscale modeling and simulation.
So on the imaging question, undoubtedly, imaging has been a big driver of the developments that we've experienced. In the patient-specific modeling, fast, low dose 360 slice CT cardiac scanning means that we can now get very high resolution patient data on cardiac anatomy very quickly and make high resolution models. And then cardiac MRI is also being much more widely used, particularly in patients with congenital heart disease where we want to follow them over time. And so we're building a database of models derived from longitudinal studies of cardiac MRI in children and adults with congenital heart disease. And I think that's going to be very helpful to physicians in managing. But also in the basic science, I think where I am most excited about imaging is imaging that sort of gets at the mesoscales that we haven't previously been able to probe. And super resolution microscopy, I think, is doing exactly that for sort of the mesoscale, that the big gap that we previously had between light and electron microscopy. And then I think the other area that's giving us a lot of structural detail that can help us scale up from crystal structures is cryo-electron microscopy. So those are just three imaging modalities that I think have all been and will be important for models in the future.
Just to add on that, like in terms of cartilage and like joint mechanics too, and there has been significant amount of efforts actually to relate the structure to like MRI type of imaging, like the GEMRI can see, like if people can understand the collagen alignments or like proteoglycan content, and from that get an understanding of the mechanical health of the cartilage, of course. But I think the other area that has emerged in the recent years, and particularly with the computational power increasing, and was the biplanar fluoroscopy has becoming more and more common, and this is essentially functional imaging, and we can more accurately measure where the bones are relative to each other, like during any type of activities, more or less. And from that information, we can understand, and get a good estimate of, cartilage contact.
So, okay. So I'm going to make a observational comment, and I'd like to hear what your feedback is on this. So with respect to the earlier question as far as sufficient level of abstraction or reducibility for, let's say, clinical effectiveness or predictability, I think those areas that have demonstrated the greatest success in this particular group are those systems that have been able to be reduced to where the primary predictive modality is a mechanical physical model in which we have natural laws that constrain those behaviors, and we have a great deal of experience with respect to the application of engineering principles to those types of characterizations of problems. And looking at the various projects, the biological components, either the cellular, molecular, genetic aspects, are essentially correlated states that can be aggregated into sets of physiomechanical properties. And therefore, the interventions can operate at that higher level because we generally aren't manipulating the cellular control structures underneath that drive those potential changes. Now, I think the need for the lower level resolution modeling is to identify whether there happen to be heterogeneities in the manifestation of those physiomechanical properties. But I think the greatest challenge has to do with dealing with those disease processes that cannot be reduced to physiomechanical rules at this point in time. And I think trying to capture the range of heterogeneous behavior associated with disruptions of cellular control mechanisms, that's where I think the next phase is that we need to go. Now, with respect to the HPC utilization within this particular field, I completely agree with Andrew, is that for a while we were enamored with making these giant models in the past, but what we actually need to do is to run tons and tons and tons and tons and even more tons of models in order to be to explore what the possible ranges of behavior that you can generate from these models that captures the heterogeneity of biological behavior. So I think that the condensation of that biological heterogeneity into mechanical physical systems is what has happened in the last 10 years with this particular group. But I think moving forward, trying to address those cellular control questions is probably where we would like to be able to move. So it's a long, rambling comment. I would like to hear what your thoughts on that are.
So I think in the interest of time, we're going to have to sort of wrap it up here. I think if the three of you have any really brief comments to that comment, go ahead and say them now, and then we'll take the rest of this out into the hallway on the break, as always.
Okay, I'll give a concise answer. I agree that the critical power that all of us who do physically based multiscale modeling are adding to the data-driven biological modeling that is also necessary is the value of physicochemical first principles in constraining the possible solutions. However, I think that applies just as much to models of molecular interactions and molecular-based therapeutics as it does to disease phenotypes that have a physical basis and maybe a physical intervention.
So let's give a final thank you to our three excellent and very different speakers and to all of you for the great questions.